drugs, or ideas, what you consume influences who you become. On the Mind and Matter podcast, we learn together from the best scientists and thinkers alive today about how your mind-body reacts to what you feed it. Before starting Mind and Matter, I spent 10 years in academia doing scientific research. I got a PhD in neuroscience where I focused on neuroendocrinology and the neurobiology of behavior, and before that, I specialized in molecular, developmental, and evolutionary genetics. I use my scientific background to help parse and translate the information that guests share on the podcast. In addition to the podcast, I write long-form written content inspired by the show where I integrate what I've learned across episodes. I also have a free weekly newsletter where I provide you with upcoming guests, share links, and provide commentary on scientific studies and research that I'm reading, and more. Visit mindandmatter.substack.com to find all of my content. Hey everyone, I want to take just a minute to tell you about a really cool health monitoring device I've been using called Lumen. It's a handheld, pocket-sized device with a sleek design. The Lumen device measures CO2 levels in your breath, which allows their technology to determine the extent to which your body is burning fats versus carbohydrates. Lumen helps improve your metabolic flexibility, your body's efficiency in shifting between using fats and carbs for energy. I use this device in the morning, or before bed, or after meals, or after workouts to track my metabolism. With just a couple weeks of use, I learned a lot about which foods were causing my body to burn mostly fat, mostly carbs, or both, as well as how long I needed to fast and how hard I needed to exercise to promote fat burning. Being able to track whether your body is burning fat versus carbs will help you figure out how your body responds to your lifestyle and whether or not your weight loss and fitness strategies are actually working. Lumen is great for anyone looking to optimize their health for either weight loss or athletic performance. Their easy-to-use app allows you to track your results together with what you're eating and how you're exercising, and it syncs with other devices like the Apple Watch. Click the link in the episode description to learn more and use the code MINDMATTER, MINDMATTER, to get 10% off your Lumen device today. That's on top of the site-wide sale they are currently running, and the Lumen device is eligible to be paid for with FSA and HSA health accounts. Check out the link in the episode description for more info. Thank you for joining me.
Wonderful. I'm delighted to share with you the mysteries of CDG.
And do you want to just start off by telling everyone a little bit about who you are and what your background is, what you study?
Yes, I have a very adventurous past. I'm a Hungarian who trained in Hungary as a pediatrician and then trained as a geneticist in New Orleans at Tulane University. That's where I fell in love with inborn errors of metabolism. And when I first heard about this rare disease, congenital disorders of glycosylation, I worked most of my career in Europe, mostly in the Netherlands and Belgium. And 10 years ago, I was re-recruited to come back to the US and work as a clinical medical biochemical geneticist, first at Tulane, where I trained, then at Mayo Clinic, and just very recently at Mount Sinai at the Department of Genetics and Genomic Sciences. I'm responsible for the training of the residents here, categorical and combined residencies in genetics, including medicine and genetics and pediatrics and genetics. And I also lead the metabolic team here and see patients and do research.
So what is glycosylation? What is the actual biology going on there under normal circumstances? What is it?
Glycosylation is a process of activating sugars, and after they're activated, to assemble them to glycan chains. These glycan chains are made step by step in the endoplasmatic reticulum and later in the Golgi. in the endoplasmatic reticulum, usually a protein accepts the glycan chain, which is synthesized and later further processed in the Golgi. But lipid molecules are also able to carry a glycan chain. So there are different types of glycosylation, and the very basic division is N-linked glycosylation, O-linked glycosylation, combined glycosylation defects, and lipid and other glycosylation disorders.
So this is a way of modifying proteins with sugars. So in other words, this is a way that our cells use sugars other than just using them for energy. They can actually sort of stick them onto proteins, and that has consequences for what the proteins do.
You hit the nail on the head. We are thinking about sugars as energy source, but we have to build structures from sugars. And without them, most of our proteins, most of our functional proteins won't be able to get their secondary and tertiary structure, and they won't be able to function correctly.
So if we think about this just in step-by-step in basic terms, we've got genes in the genome, in the nucleus of the cell. Those genes are transcribed into RNAs. That's the process of transcription. And then those RNAs are translated into proteins and folded into proteins. But it doesn't necessarily end there. Basically, one of the things that can happen after that, a post-translational modification, is this phenomenon of glycosylation. Basically, a chain of sugar molecules is essentially stapled onto the protein, and that helps it do stuff.
Yes, I think you described it quite well. I think that the structure without the sugar antenna would be very different. And for example, in a case of an enzyme or a transport protein, if that glycan is not attached to the protein, then maybe the active site remains open, or just the configuration of the protein would be so different that it won't be able to carry something like iron or copper.
Yeah, so it has potentially a very strong effect on what the proteins can do and how they work, whether or not they work as intended. And I think you said that, did you say that most proteins have some kind of glycosylation? It's a very common thing?
Actually, most functional proteins do. I can give you examples for such proteins. Secretory proteins in measurable in your blood are very good examples. And actually, a good example is coagulation factors. Almost all coagulation factors are glycosylated. And if you miss the glycan, then you don't have normal clotting. So you have a surgery and you would bleed, or you have an accident and you won't stop bleeding. Also, anticoagulation factors are glycosylated, so even more complicated. In a problem with glycosylation, you can bleed and also get a stroke or a thrombotic episode because the clot, whenever it forms, it cannot dissolve. But other good examples are hormone regulatory proteins. If you think about proteins which are essential for your survival, FSH, LH, for example, in your brain, which is essential to get you into puberty, or thyroid stimulating hormone, which is essential that your thyroid gets enough stimulation to keep you in balance for your energy regulation. Other examples could be transferrin, which is transferring your iron in your blood. But it seems like I only talked about secretory proteins. You should know that immune proteins are also highly glycosylated. So immunoglobulins, you won't be able to fight bacteria if you don't have glycosylation, because that's the actual chain which recognizes or surrounds the bacteria for further killing action. And then all your proteins in the brain, which are, for example, on your cell surface to signal to other cells, very often glycosylated proteins. Receptors are almost always glycosylated. So your acetylcholine receptor in your muscle, if you don't have the antenna, it cannot receive the acetylcholine message and cannot contract. So it's quite complex. If you don't have glycosylation, you are involved every little piece of your body, every inch is affected by a glycosylation problem.
Wow. So lots of different proteins are glycosylated. This affects all sorts of stuff in biology. I would imagine, so if glycosylated proteins are involved in everything from the immune system to metabolism to development to, muscle and neurons. Yeah, I would imagine that if there's a problem with the glycosylation, that lots of different things could go wrong.
So I told you that there are different glycosylation sorts, right? And glycosylation is the most common pathway, which is affected by human disease. And you cannot really mention an organ which is not affected if you have a congenital disorder of glycosylation. So all organs get some functional defect in most of the common N-glycosylation disorders. And there are just a few exceptions where only a few organs or just the brain is affected. I have to add to this, though, that I mentioned all glycosylation disorders, and these are more tissue-specific disorders. Mm-hmm. So it's not so easy to diagnose them because you don't have a blood marker because N-glycosylation mostly happens in the liver, so you secrete out the proteins and then you can measure them in the blood. But in all glycosylation disorders or lipid-linked glycosylation disorders, you need very specific tests because the liver is not so much affected and not giving us easy markers to diagnose the disease.
Mm-hmm. So given that glycosylation is the addition of sugars, chains of sugars to proteins and things, does dietary carbohydrate intake affect glycosylation?
Well, you made a big jump going towards potential therapies. It's a very important thought, and there are a few disorders. Maybe I should mention there are actually 200 disorders known due to glycosylation defects. So that's really a high number of type of disorders under one biochemical umbrella. So from these disorders, unfortunately, only maybe 6 or 7 are treatable by dietary modifications. And these could be adding very special rare sugars to the diet or adding certain elements like manganese or magnesium, which are very important for the normal function of glycosylation, mostly the Golgi, but in some degree in the ER as well.
Mm-hmm. Mm-hmm. So after proteins get made, they're often being glycosylated within organelles like the endoplasmic reticulum or the Golgi apparatus. When this is happening, I want to understand a little bit more about the mechanics of glycosylation. For the actual process itself, is it an enzymatic process? Are there dedicated enzymes that are attaching or doing this to proteins? How does it actually happen? How does it get stuck? How do the sugars get stuck to a protein?
Yes, you're right again. And if you ask me how I teach this to my students, I usually say it's like the ER is a conveyor belt, and lipids like dolicol are the actual seats where glycosylation starts, and an enzyme comes and grabs a sugar. And then the next step is another enzyme comes and add another sugar. And there are many, many steps just along the ER. There are 20 something steps which happen enzymatically. And then you go to the Golgi, and there are very specific transporters, which are also enzyme-like proteins. They bring in special sugars to the ER, but there are also the conveyor belt is working in the Golgi as well. So you have enzymatic steps following each other.
Mhm.
Now, you asked me, maybe is that the only way you can get a congenital disorder of glycosylation? Actually, there are several proteins which are responsible for Golgi integrity,
Mhm.
trafficking regulator proteins, for example, which are not per se enzymes and have more global problem or global function leading to glycosylation problems. You could say maybe these are secondary glycosylation disorders, but at the end, these regulators affect the enzymes in the step-by-step or stepwise process. So from my perspective, these are also true glycosylation disorders.
Mhm. So I would imagine that a congenital disorder of glycosylation means that there's just some problem with this process somewhere in the assembly line. There's probably multiple places where things can go wrong, but because this is such a basic and widespread cell biological process, there's many proteins, as you said, that get glycosylated, and so this has, it's a widespread thing. It's not just an isolated thing. I would imagine that when you have a problem in the glycosylation assembly line, so to speak, it's probably the disease probably manifests in a very general way. It's not like one part of the body is affected. It's probably a whole body issue. Is that the way these disorders work?
For N-glycosylation disorders, this is true for most N-linked glycosylation disorders. It is probably surprising for you that in O-linked glycosylation disorders, sometimes only one organ or a few organs get affected. For example, there is an O-glycosylation, O-monosylation disorder called muscle eye brain disease. And it's a very severe disease, and patients can be born without eyes or very tiny eyes.
Hmm.
Their brain can be so malformed that they don't have gyri, so
Hmm.
they miss the gyration of their brain
Oh, so the
and
brain is just like smooth.
smooth,
Wow.
exactly. And you can imagine you cannot really learn much if you're born with a brain like that. And then the muscle is affected and weak and gets weaker with time. So we still don't really understand why other organs are not affected. We understand why the organs or systems affected. Why do they get affected? Because the monosylation is very important. For example, for a muscle protein called dystroglycan. So I understand why then the muscle weakness happens, but why other organs do not suffer, why it's not expressed in all the organs. That's an exciting question for the future geneticist.
Hmm, hmm. And you said before, you said something about special sugars being involved in the glycosylation process. Are when a protein, say, is glycosylated, these these sugar motifs that are stuck out of the protein, are they made out of like special sugars, or are they composed of things like glucose that we think of as sugars normally?
Yes, that's a very important question. And if you think about biochemistry, I think that it's clear that glucose should be used for something else. And some glucose molecule, how should I say? So, for example, N-acetylglucosamine, that's a sugar which is built into the sugar chain, but glucose itself, it doesn't. It has a regulatory role, but we are not spilling our glucose to build it
Hmm.
in structures.
Yeah,
But
yeah.
there are a couple of very special sugars. One is, for example, mannose, which is very important in the sugar chain, especially in the brain. And that's a sugar maybe you heard about because when somebody has a UTI, then
Mm.
this is prescribed to help urinary tract health,
Hmm.
and also available in cranberry and other sugars, but it's very, very tiny amount
Hmm.
what's in the diet. So actually the body keeps these sugars inside and not spill them. So actually you reuse them for your glycosylation. You don't burn it, you
Uh,
don't pee it out, you
yeah.
just rebuild it.
So there's different sugars in the world. Basically, it sounds like what you're saying is from like a bioenergetic and efficiency standpoint, glucose is an energy substrate for our cells. So we want to use the glucose for energy purposes to make ATP. So we're generally not going to use that to build these sugar residues that are going to be part of the glycosylation process. We use other types of sugars. Are all of those things dietary in origin, but we just ingest them in smaller amounts, or are we sort of making new sugars from other sugars or something?
Yes, you can actually make these new sugars. So, for example, phosphate can be made from fructose.
Mm-hmm.
So you're able to make them, but they are also in the diet. But there's such a small amount that usually you just keep them inside, and then when protein gets old, the sugar residues they get cut off, and then you get back all your substrates, right?
Hmm.
And you break down your protein and you start to build a new one, and then you make a new antenna.
I see.
So you vary, you know.
So there's a recycling process when a protein gets made, maybe it gets glycosylated, it does its thing for a while. At some point, it gets old, it's broken in some way. The cell rips off the sugars, probably breaks them down and reuses them, and sort of the whole process can happen again with recycled components.
Yeah, very frugal, right? This is just reuse everything.
Hmm.
And other rare sugars, maybe an important sugar is galactose, which is really in the diet, right? That's
What was it called?
galactose.
Oh,
So that,
galactose. Okay.
yeah, galactose. And I'm Hungarian. I'm allowed to pronounce
It's
it
okay.
as I wish.
I actually visited Hungary last year. It was a really cool place.
Yes, it is. Budapest, especially. So galactose is the monosaccharide from lactose, which is in our diet, and we think it has a very important role. When you are born, you get a lot of mother milk, and actually, that's how you fill up your galactose reservoirs. And patients with significant galactose restriction could develop some problems just by the dietary restriction. That's how important that sugar is for
Wow.
your biochemical health.
Interesting. So that sugar is found uniquely or mostly in milk, and if you were to restrict it too much, that could lead to glycosylation issues, basically.
Yeah, secondary, yeah. But actually, it's also in tomato and in some vegetables. So it's really funny,
I
probably.
see.
People don't know that it's also in vegetables, but
Yeah,
most
yeah.
dairy.
And because these sugars are minor sugars, we don't consume them in large quantities like we do with glucose. And because these recycling mechanisms exist, you kind of just need a little bit in your body. It sounds like your cells can sort of hold on to them, store them, and recycle them for long periods of time.
That's correct.
And so you've used the analogy of an antenna multiple times. A lot of times these glycosylation residues are acting as something like an antenna, helping a protein, sense something in the environment.
Yes, maybe antenna is not so good idea, although in the brain, I'm thinking about, for example, lipid-linked glycosylation, these stick out from the cell surface, right? And signaling is important. But also you could say arms, because when immunoglobulins have their sugar chain residues, they actually find the bacteria and come around the bacteria and sort of grab it.
Oh
And
wow.
then the killer cells come in and they kill the bacteria. So coagulation, if you think about it, like a coagulation factor has the sugar a chain attached to it, then another coagulation factor comes, and these sugar chains, they help to assemble the different factors and make the clot. So protrusions, or it's
they're attached to the proteins. They stick out usually, and they're doing something functional. I mean, oftentimes when we talk about biology, we think of sugars as being for energy, and we think of proteins as being for doing stuff. But what you're saying is actually certain sugars can get attached to proteins and be part of the doing stuff.
The action.
Yeah. And so what are some of the more common, I mean, my understanding is these things are all pretty rare, but among them, what are the more common congenital disorders of glycosylation, and how do they clinically present?
So the most common type is called PMM2 CDG. And you have to know that we name these disorders based on their gene. So the defective gene name, and then we add CDG. So PMM2 is from phosphomonomutase, and the gene for that enzyme is PMM2. This is a disease which was described by Professor Jaken, who's a famous Belgian pediatrician. He's still alive, and he still sometimes sees patients in Leuven, Belgium. The story is really, really fun to hear how he discovered this, because this was 45 years ago. He encountered two patients who had a bizarre number of blood abnormalities. The patient had bleeding abnormalities, but then more than one clotting factor was wrong, and also problems with dissolving the clot. Had hormonal problems, but not just the thyroids were wrong, but there was also the prolactin, which is important for lactation, but also sex hormones were abnormal. And then their transport proteins as well. And so it was just a bizarre picture. Nobody saw anything like that. And then twins both had the same presentation with some developmental delays. And so he was just thinking about what's common in all these proteins. And he just said, well, they are all glycosylated proteins. So there should be a new genetic defect of disorder of glycosylation. And so it was more than 20 years that the gene was found, and he was right. But it's just showing that some clinicians are brilliant in discovering disorders before we have the way to prove. And but that was also, or it gives an answer to your question. So most patients with this disorder have all these really bizarre clinical picture with bleedings and thrombosis early on, liver problems and endocrine problems, low blood sugar, and not going into puberty and not growing. And that's only just a few symptoms I mentioned, because obviously other organs can be affected: the heart, the kidney, the muscle.
The clinical presentation is multifaceted, and that's what makes it bizarre. They've got multiple issues with blood clotting and or developmental problems and or multiple hormones that are not acting appropriately. So it's not like there's just one process that's defective or one protein you can point to, which is probably what made it so mysterious. But it also speaks to the fact that this is a general process. If you've got a glycosylation issue, it's going to affect lots of different stuff.
Yes, you're completely correct.
And so how common is this? Is this extremely rare? Is it like how rare is it?
So this disorder I mentioned, PMM2 CDG, it's about 1 in 20,000. So actually it's, of course, it's a rare disease, but this is a frequency we would do newborn screening for if we had an FDA-approved therapy. But unfortunately, we don't, not yet.
And so what are people working on in terms of possible therapies? Is there anything that looks particularly hopeful? Are there I would imagine that there's, potentially some drug therapies people are working on, but also earlier you mentioned the possibility of dietary interventions. This is a biological process that involves sugars, using sugars. Is there a way that it can be affected in some way by diet?
I would like to bring up another disorder. It's less common, but just maybe easier to understand
Mm-hmm.
the treatment dilemma. So one step before this enzyme defect is another defect where it's called phosphomonoisomerase. And that's the first phosphorylation of or phosphorylated monos, which is in this machinery of activating monos, the rare sugar, which is essential for the glycans in the ER. So in that case, because phosphorylation of a sugar at the sixth position of the sugar molecule is possible by another enzyme, you can treat it with giving a very high dose of monos in the diet. And then that monos will trigger the hexokinase, which is normally phosphorylating glucose, to phosphorylate the monos. But you have to give a really high dose.
Mm-hmm.
So it's almost a diabetic dose of monos you're giving to the diet.
Wow.
And then
So normally there's so much glucose and so little mannose that this enzyme sort of will never just see it and process it. But if you just really crank up the mannose relative to the glucose, it will actually work on it.
yes, exactly. And I just wanted to tell you what's the name for this. This is like a fun name for this enzyme. This is a moonlighting enzyme because it comes in and helps out.
Yeah, it has a
Yeah. Yeah. So that is possible in this MPI CDG phosphomonoid isomerase defect. But the step after, which is actually the most common CDG, PMM2 CDG. Unfortunately, that is coming a phosphor to the first place, so the monos molecule at the first position, and there is no other natural enzyme which could take over that task. So it's a very difficult challenge because options for treatment would be either you provide this phosphorylated monos to the body, but then it's an active phosphorylated molecule. It wouldn't make it to the cell, wouldn't make it to the right compartment, but also wouldn't so unstable, but also would hurt membranes. Other option is to change somehow the configuration of the sick enzyme. So use a chaperone or use small molecules which would just change the configuration that the enzyme could be more active. Or you could also think about genetic treatments like gene therapy, right? So there are options, but none of them are trivial and easy to accomplish because it's a very specific compartment of the cell and hard to direct the therapy there deep inside.
Yeah, yeah. And when patients have a congenital disorder of glycosylation, I mean, in general, what are the outcomes? Is this something that affects lifespan? Can they live somewhat normal lives? Are they like very much impaired?
The initial disorder was a very severe disorder described by Professor Yakin. The twins discovered then are 40 something year old. They're still alive and they're in wheelchair, but they're able to stand up and make a few steps. They have some speech. They are very happy and very social individuals, but they do need a lot of care. This is the severe end of the spectrum.
Mm-hmm.
And since I'm seeing many CDG patients as it became my focus, and I really enjoy meeting the families and taking care of the kids. I met exceptional mild cases diagnosed cases where the diagnosis was made at 18 years based on delayed puberty and no other symptoms. I have little ones who are developing pretty much close to normal. So far, so good. We don't know if that stays that way, but I think that the phenotype is quite variable, and there are many. Oh, and the oldest patient is 76, I know of, and that's also good news for outcome prediction.
And so so in the field, your work, or just anyone in the field, what are people working on today as it relates to developing therapies?
Drug repurposing is very strong in the CDG field because chaperones or small molecules could really benefit enzyme activity, and not just in PMM2 CDG, but in many other disorders. This is a viable option. Besides that, there are several CDGs where there is gene therapy already on mice or fish model level. And there are 2 or 3 gene therapies which are in clinical trial for CDG.
Mm-hmm. And I would imagine, if say, a CDG exists because there's some sort of enzymatic defect, a mutation in a gene that's involved in the glycosylation process, you would want something like a gene therapy in order to fix that, and you'd want to probably fix it early. So right, there's probably a developmental aspect to this that's important, because the defect in glycosylation, if it's there from the beginning, it's going to impact the entire trajectory of development. So in theory, you'd want to sort of correct the issue early on in development, I would imagine.
Yes, I should interview you because you're so good in glycosylation. You would get an A+ from me. Yes, you're right. This is a big challenge. Glycosylation is so important for fetal development and cell migration that even though the mother compensates some degree, but we do see malformations, especially brain malformations, cerebellar malformations in many CDGs. And so if you want to treat, then the optimal treatment is to diagnose these fetuses early and then maybe treat them intrauterine. That would be the coolest thing.
And so you said this is about 1 in 20,000 people or something like that. So when you have something that's this rare, it's common enough that there's probably quite a few people out there that have this in their lives, but it's less common than other things. How does that affect the ability to obtain funding for this research? Is that a challenge?
Yes, it is a challenge. I think rare disorders, just go back to the frequency. So you asked me the most common disorder, PMM2 CDG, 1 in 20,000. Some disorders are extremely rare, 1 in several millions. So there are really unique CDGs as well. You can imagine even more difficult to get financial support for such a disorder. But from my perspective, glycosylation is so important in so many, specialty. If you just think what I told you, from hematology to immunology, but even in cancer, because glycosylation is essential for metastasis. So if you use your CDG as a model to understand glycosylation, then you can, deduct certain things which could help common diseases as well. Yeah. So hopefully one day people understand that and we'll get a lot of FDA support.
Yeah. I would imagine in your field, even though you're focused to some extent on the CDGs per se, understanding glycosylation as a biological process has ramifications that go beyond CDGs.
Exactly.
Yeah.
Exactly.
Yeah. And then
we just need to teach that to the NIH and all the authorities very timely.
Yeah, I bet. So because this is such a basic aspect of biology, glycosylation is so common. There's so many different proteins that get glycosylated. What are there? So if you don't have a CDG, if you're just an otherwise normal person, are there defects in glycosylation that can develop as a consequence of, say, just metabolic dysfunction or chronic disease? Can something go wrong with glycosylation in a normal person, basically?
Yes, this is a significant problem if you have a liver disease.
Mm.
Now it's a pretty hot topic, NASH. I don't know if you know that non-alcoholic liver disease got designation like that. And that's just one example. Actually, autoimmune liver disorders and other liver dysfunction also have a major effect on glycosylation because that's where it's happening. And glycosylation, more than 90% of glycosylation happens in the liver.
Hmm.
So that's a good example where it's changed. And the other is cancer because the glycosylation changes, so metastases can spread. In my patients, actually, cancer is very rare. They are missing glycosylation, so cancer, in case a cancer appears, it doesn't have that easy way to spread to other parts of the body.
Mhm, mhm. So if I'm hearing you correctly, a lot of glycosylation happens within the liver, and if one develops non-alcoholic fatty liver disease or fatty liver from any cause, that probably then starts to compromise the liver's ability to glycosylate things normally.
Mm-hmm. And then that depends on how severe the liver disease is, but in a case of a severe liver disease, glycosylation will get affected.
Mhm, mhm. What is it, this is a basic cell biological process. It can manifest in these diseases, these CDGs, but glycosylation is important for everyone. The biochemistry gets pretty complicated pretty fast. I mean, if you had to sort of summarize everything, what do you want people to take away from this conversation in terms of the importance of glycosylation and the need to study it?
Well, it's a big ask. Um, so I'm a clinician and I mostly focus on my patients.
Mm-hmm.
And I really focus on trying to understand how glycosylation affect their function and and to try to treat that. So that's my personal interest in this. But evaluating glycosylation, we learn so much about the function of the body that there is no student or trainee who wouldn't benefit understanding this more because you can just take that information and use it as a diagnostic method or understanding pathobiology or changing proteins in their function by altering glycosylation. So I think that that is really relevant for everyone. And another interesting a little anecdote, PMM2 CDG patients are genuinely cheerful and social.
Hmm.
I'm not saying that there are no exceptions where a child has severe developmental delay and autistic or learning problems, but if you one day meet the kiddos I'm taking care of, you would be surprised how they embrace life and that sunshiny attitude is actually contagious. Um, so what I want to learn one day, what is the genetic or biochemical reason for an uncomparable positive attitude in a severe multi-system disease compared to other
Hmm.
metabolic disorders where the patients are so frustrated and unhappy and yeah
Yeah.
different.
They're often cheerful, or you might even say, I don't know, depression resistant.
Yes, so there are exceptions,
Mm-hmm.
but majority of the patients are so lovely. It's hard to resist.
Interesting. And I don't want to take too much more of your time because I know you're busy and you got to get back to the clinic. Is there anything you want to reiterate from this conversation or any final thoughts you want to leave people with about these diseases or about the underlying biology?
You want to come and train with me? I think you'd be a great trainer in genetics. Are you interested in further developing your career and be a geneticist?
Well, actually, my original training was in genetics. That was in my bachelor's degree. It's been a while since I've been in that world, but we'll see what happens.
Mhm. Maybe the only thing I want to say is that CDG research is heading towards developing good models that we can better understand what's happening in the human body. And we are starting to leave animal models like fish and mice behind. And one exciting perspective is the use of organoid systems, which we develop from patient-derived cells. That's brain organoids, heart organoids, liver organoids, and 2D or 3D systems. And then if you think about it, you have the whole epigenetic, genetic, everything environment in your little, model system. And so if you find a treatment in that system, that's very easily translatable to that individual. Even it's ASO therapy or gene therapy or a small molecule. So I think that for glycosylation research, this is really a breakthrough because most of the animal models do not survive the genetic. If you build these models, then the mice either doesn't show the phenotype or dies, not even born. So there is a new era for us, and I think these organoid and patient cell-derived model systems are really cool to make the next step and understand this better and then to find therapies.
All right. Well, Dr. Eva Morava, thank you very much for your time.
Thank you.
This episode is supported in part by Athletic Greens. Their main product, AG1, is a comprehensive and convenient daily nutrition product containing 75 vitamins, minerals, and whole food sourced ingredients with less than 1 gram of sugar per serving, no nasty chemicals or artificial anything. It's gluten and dairy free and compatible with paleo, vegan, vegetarian, and ketogenic diets. AG1 is a quick and convenient way to supplement your diet to help ensure your body is getting the nutrients it needs. It comes in powder form and you can mix it in water and drink it, or you can put it into a smoothie or a shake or something like that. I mix it into water and drink it, and it's super convenient. If you go to athleticgreens.com/mindandmatter, Athletic Greens will give you a free 1-year supply of vitamin D and 5 free travel packs with your first purchase. Their vitamin D product comes in tincture form, so you just take 1 drop each day. A large fraction of the population is actually vitamin D deficient, especially in winter months when we get less sun exposure, and vitamin D is super important for the proper function of the immune system and for a variety of other things. So visit athleticgreens.com/mindandmatter or click the link in the episode description. You'll get a free 1-year supply of vitamin D with your first purchase.